Welcome everyone. What we're going to find in these PowerPoint slides is just an explanation of some probability with our genetics and examination of the chi-square. That is how we evaluate the difference between what we observe versus what we expect. We did this in lab during weeks 1 and 2, but I thought it would be helpful if you also had a resource to come to and review what you need to know for probability as you prepare for the exam. So let's go and get started. So let's think about some basic probabilities. For instance, what's the chance of pulling an Ace of Spades from a deck of cards? Well, we know there are 52 cards in a standard deck of cards, and there's only one Ace of Spades, so it would be a 1 out of 52 chance. Chance of pulling a face card. Well, we know there are how many face cards? There are 12 face cards, so it would be 12 out of 52, and of course you could reduce that down. When you determine these probabilities, for the purpose of this class, you can express it as a fraction or as a decimal. I want to first look at the multiplication rule. And if we look over here at this picture here, what's the chance of taking a single die and rolling it and getting a 4 on it? Well, we know that answer to be 1 out of 6 because there are 6 sides to the die, and as long as it's a standard die and it's not weighted in any way to make it a trick die, we know that there's 1 out of 6 chances that it'll end up on a 4. Now it gets a little bit more complicated, but not too bad. If we ask to get a 4 on 2 sequential rolls, what would that be? Well, to do this, we have to take the multiplication rule. And we know on that first roll of the die, it's 1 out of 6. On the second roll of the die, it's also 1 out of 6. There's no increase or decrease probability of getting a 4 on that second roll. So we multiply these two separate probabilities together and get a 1 out of 36. So whenever you see these, the word "and" is key. If you see the word "and," you know we're talking about the multiplication rule. And you know that you're going to multiply the two probabilities. So to get a 4 and a 4, you would multiply 1/6 by 1/6. In order for the multiplication rule to apply, each independent has to be separate and independent. So having a girl baby and then a girl baby. The second event is not dependent on the first event. All right, let's move on to the addition rule. The addition rule applies to get the probability of one of 2 or more mutually exclusive events. You must add the probability. So let's use the rolling of the die as an example. Probability of getting a 3 or a 4 on the single roll of the die. So you can get either one of those two. We know each individual event would be 1/6, but since it's the same roll of the die, we add them together. So 1/6 plus 1/6 equals 2/6 or 1/3. All right. So these are mutually exclusive events. Now, this kind of sounds a little silly. Probability of having either a boy or a girl. Well, we know it has to be a boy or a girl. Probably not the best example, but it does fit because you know that each one of these are mutually exclusive. And so for that answer, it would have to be 100%. So you cannot throw a single die and get both a 3 and a 4. It's got to be one or the other. So now let's look at some pea plant crosses and apply the multiplication and addition rule. So even though the Punnett square is probably the easiest thing to use for a monohybrid cross, as we're introducing this, let's think about replacing the Punnett square by just doing these simple probability rules. So if you do this cross, big T little t times big T little t, and you ask what's the probability of either parent producing gametes with big T or producing gametes with little t? That is, what is the probability of getting a big T progeny? Would be another question you could ask. So the first thing you'd have to ask is what is the probability of a T gamete, a big T gamete, and a big T gamete from these parents? Well, the probability that this parent here is going to produce a big T, or this parent producing a big T, is 50%. Because we know, based on Mendel's rules, that these two alleles during meiosis, they're going to separate it from each other in equal probabilities. So we know there's a 50% chance of getting big T for each of these two parents. So we take 1/2 times 1/2. Again, these are independent events. What's the probability of this parent producing a big T gamete and what's the probability of this parent producing a big T parent? So we multiply them, follow the multiplication rule. We could get the same answer for big T, little t, big T, and little t, little t. It turns out to be the exact same because of Mendel's rules in this example. So what is the probability of getting a tall plant? This is a little bit more complicated, but not too bad. So in essence, we have to ask, what's the probability of getting a big T, or a big T, or a little t, big T? And we know that there's a 1/4 probability, 25% probability of getting a big T, just like we saw here. Or the probability of getting big T is 1/4, or the probability of getting a little t, big T is also 1/4. And so when we add these together, because again, these are OR events, we can't have all three of these happening at the exact same time, so they're mutually exclusive. So we add them together and we get 3/4. Now, of course, if we did a Punnett square over here on the side, you would see it's the exact same, we'd get the exact same results. All right, so let's consider this trait here as an example using albinism. We know albinism is a recessive disorder. We know that individuals who are big A, or big A, little a, they have pigmentation. Someone who is little a, shown here, they have albinism, such as we see in this picture here. Two parents, both who are big A, little a, have what probability of having a child with albinism? We know that answer is 1/4. You can do a Punnett square to figure that out, or you can ask the question is, what's the probability of one of the parents producing a gamete that has little a, and what's the probability of the other parent producing a gamete with a little a? And you multiply those together and you get 1/4. With pigmentation, what's the probability? Well, we know to get pigmentation from two parents that are heterozygous is that you'd have to be either big A, little a, or little a, big A. Same as we saw on the last slide for tall plants. So it's 3/4, because it turns out to be 1/4 plus 1/4 plus 1/4, using the exact same rules we saw on the last slide. Now, what's the probability, to make it a little bit more complicated, what's the probability of having 3 children, each with albinism? And for this is that we know that the first time the parents have a child, there's a 1/4 probability that their child will have albinism. Now, if they have another child, nothing changes. There's still a 1/4 probability. And again, if they have a third child, there's still a 1/4 probability. So we multiply these together and we discovered that there is a 1 in 64 chances that they will have 3 children, each with albinism. Again, we're using the multiplication rule here. Okay, so let's look at a more complicated problem. What is the probability of these same parents, the same heterozygous parents, big A, little a, having 3 children? But this time we're going to ask, one of them has albinism and the other 2 have pigmentation. I'll give you a hint. The answer is not 9/64. Now, go ahead and, as you're watching this video, hit pause and try to figure this out. And then hit play again, and you'll see what the answer is. This is a good practice. Okay, so you have to ask yourself, what is the different possibilities of having these 3 events occurring? The first possibility is the first child will have albinism and the next 2 children won't. Next possibility is that the first child will not have albinism, the second will, and the third will not have albinism. And the last possibility is that the first 2 children will not have albinism, but the third will. Each of these 3 events here, 1, 2, and 3, are mutually exclusive. Any one of them could happen. We don't know which one could happen, but we know only one of these 3 can happen. So because of this, we know that each of these has a 9 out of 64 probabilities of occurring. We know that there's a 1/4 chance that this could occur, and we know there's a 3/4 chance and a 3/4 chance. So you multiply 1/4 times 3/4 times 3/4, and you end up with 9/64. But since each of these 3 events could occur, and each of them is equally likely, we add 9 64ths plus 9 64ths plus 9 64ths. So the correct answer is 27 out of 64, as shown here. It's very easy to forget about these different possibilities and just quickly put 9 64ths, but that wouldn't be correct. You have to put 27 64ths. This is a good example of how we would utilize both the multiplication and the addition rule in the same question. All right, now let's take even a more complicated problem. Same parents, heterozygotes, big A, little a, they have 5 children. And these 5 children have two have albinism and 3 have pigmentation. What was the probability that they would have these children? That is, what is the probability of having 2 children with albinism and 3 with pigmentation? So this is a little bit more complicated. You can try to, like we did on this slide here, figure out all the different possibilities that this could occur, or you could do our good friend binomial expansion. Binomial expansion is p plus q raised to the n. In this, p is the probability of one event occurring. Let's say, for the sake of argument, that we're going to say p is the probability that albinism will occur. And q is the probability that the other event will occur. And in this case, let's say it is not having albinism. N is the number of times. In our problem here, the number of times is going to be 5. So for this problem, it's p plus q raised to the 5th power. And if we break this down, we have p5, and then I'm not going to read these all to you because you can read, you can read just as easily as I can. But what these mean is that this first one, p5, means what's the probability of having 5 children all with albinism. The next one is p4q, what's the probability that 4 children will have albinism and 1 child won't, because remember, even though it's not stated here, there is a 1 here. And this 5, all this means is that there are 5 different possible ways that you could have 4 children with albinism and 1 without. Same thing here. P3Q2 means what's the probability of having 3 children with albinism, 2 without. And then we multiply it by 10 because we know that there are 10 different ways that you could have this combination of children with and without albinism. Okay. So on the exam, I should tell you that you will be given this equation, and you will also be given this Pascal's pyramid here. And we use this pyramid, and the way we use this pyramid is to assign the numbers that go in front of these probabilities. So in this case, 1, 5, 10, 10, 5, 1. And we use this row here. And you know you need this one because there's 6 numbers here, and we have 6 different equations here. We know if we added these did all these calculations together, that our answer would be 1. Okay. So let's work our problem. In this particular problem, what we have to do is 10p squared q cubed. All right, and that's, where's that one at? It's this one right here. Again, because the question asks up here, what's the probability of having 2 children with albinism and 3 without? So p squared, q to the 3rd, and we multiply it by 10 because there's 10 possible ways to do this. So we know that the probability of having a child with albinism when both parents are heterozygotes is 1/4. We square that, and then we know the probability of having a child with pigmentation is 3/4, so we raise that to the power of 3. When you do this, and then you of course multiply it all by 10, and when you do all that, you get an answer of 270 over 1024, or you could write it as 0.26, or you could write it as 26%. I'd accept any one of those 3 answers. Now the key to being able to do this problem is being able to take this simple equation here and breaking it down into this. And if you need help with going from this step here to this, make sure you come see me and I'd be happy to kind of walk you through it. On the exam, there will also be this equation given to you. This is an alternative formula. I'm not going to do a problem with it, but it gives you the exact same thing. Go ahead and work through this using this equation with the problem on the last slide and see if you get the same answer of 0.26. And on the exam, you're welcome to use either this formula or use the binomial expansion. Either way is fine with me. All right, let's move on to this last part, chance, statistics, and our good friend, the chi-square. So based on Mendel's laws, we expect to see certain ratios of progenies, 3 to 1, 9 to 3 to 3 to 1, and many others. But we know when we do an actual experiment, we will see some deviations. When we do a cross, we don't expect that, well, if you say you get 100 flies from a cross, we wouldn't expect that exactly 75 of them will have a certain trait and 25 will have the other trait. We might get lucky and it may turn out that way, but we know because of chance that this isn't always going to be the case. And so what we want to know is there has to be some kind of test to determine if what we get is close enough to 3 to 1 to say that it is due to chance. The difference is due to chance. Okay, so deviation is expected to be less as the sample size increases. So for instance, if I take a coin and I flip it 2 times, we would expect that 50% of those times it'll be heads and 50% of the time it'll be tails. But if I just flip it 2 times, you wouldn't be surprised if both times it ended up on heads or ended up on tails. However, if I flip that coin 1,000 times, we would expect to see a closer 5 to 500 of each heads and tails. We would be very surprised if we flipped it 1,000 times and 950 of those times it was either heads or tails. Okay, so how do you know if your data that you have observed fit your hypothesis of your expected ratios? Again, you did this in lab. In fact, you did a lot of these in lab, but this is just another source for you to look at. So again, we're going to use the chi-square test. So let's look at this example. We're doing a cross between these 2 heterozygotes. And if a question says monohybrid, this is a good clue to you that what we're talking about are 2 heterozygotes being crossed. If it says test cross, we know what we're looking at is one of the parents is going to be homozygous recessive. In this case, it would be little b. So keep that in mind. But this is a monohybrid cross. And for the most part, what we're going to see is if it's a monohybrid cross, our prediction will be a 3 to 1. All right, you do this cross, and you get 30 black cats and 20 gray kittens. And our problem here tells us that this B allele gives us a black cat, and when you're homozygous recessive for B, you get gray. So what is the expected? Well, we would expect if we add these up, 50 cats here, and if you take 50 and you multiply it by 75%, you would expect to get 37.5 cats as being black, and then you take this number of 50 of total cats and you multiply it by 25%, you would expect 12.5 cats to be gray. Now, obviously, we don't expect a half of a cat to be black or half of a cat to be gray. These are just our expected numbers. Our observed numbers, of course, will be whole numbers. So, is this difference that we see, 30 black to 20 gray, in our observed data, and if we compare that with our expected data of 37.5 and 12.5, is this difference due to chance alone? Or is there something else at play here that's allowing for more of either black or gray cats to occur than we would expect? So to do this, we do the chi-square test. But let's remember, there's a few things the chi-square test tells us, but there's a few things that the chi-square test does not tell us. For instance, the chi-square test does not tell us if the genetic cross was done correctly. So if you made a mistake and accidentally crossed 2 black cats as opposed to say a black cat that was big B with a black cat that was big B, little b, the chi-square isn't going to tell you that. That was just the way you set up the cross, and it happens to be wrong. So it also tells you it does not tell you if the results were correct, and it also does not tell you whether we chose the correct genetic explanation. Maybe our prediction should have been something other than 3 to 1. All we're testing is, does it fit our prediction of 3 to 1? So it does tell us the probability that the difference between observed and expected is due to chance alone. So let's work through this problem. We will always have to state a hypothesis. If you always state your hypothesis just as I have written here, you will be in good shape for the exam. So in this case, the deviation between the observed data and the expected ratio of 3 to 1 is due to chance alone. Then we need to determine what our expected values are, and we've already done that, 37.5 black cats and 12.5 gray cats. Again, we got that by adding the total cats, which was 50, multiplying it by 75% to get our dominant, and multiplying that total by 25% to get our recessive. Then we plug the data into our chi-square formula, which we have here. Determine if the null hypothesis is either rejected or not rejected. And if we reject it, we say the deviation between the expected and the observed is not due to chance alone, and so we have to propose an alternative hypothesis. If we do not reject it, we have to say our observed deviations can be attributed to chance alone. Okay, so here's our chi-square formula that you've worked with quite a bit already. O is equal to your observed values, E is equal to your expected. Chi-square is the sum of the calculated values for each of these for each category of the ratio. All right, so once chi-square is determined, it is converted into a probability, a p here, and using the degrees of freedom. Again, degrees of freedom is n minus 1. N is the total number of events possible, or categories possible. In our case, we have two possible categories, a black kitten or a gray kitten. So in this case, it's going to be 2 minus 1. And in our problem, the degrees of freedom will be 1. All right, so let's plug our numbers in. Our observed is 30. Our expected is 37.5. And for black cats, and then for gray cats, it's 20 and 12.5. We then take this, we subtract them, subtract 37.5, and we square them and divide it by our expected. In this case, it's going to be 37.5. And then we do that with our category for little b, and then we add them together. So if you're not comfortable doing this yet, based on the practice problems in lab, go ahead and work this through again in your own format here, and make sure you get the same answer. But when we do this, we get a chi-square value of 6. And our degrees of freedom, as we said on the last slide, is 1. And then we're going to use the table 3.4 to determine the probabilities. Again, the table such as this one will be provided in the exam, on the exam. So the only column we're going to look at here is this one, or I should say the only row we're going to look at is this one, because it's where degrees of freedom 1s are. And we're going to look and see where our number 6, our chi-square value, remember, is 6 here, where that falls. And we can see it falls right in between here. And so what we'll say is there is between a 1 and 2.5% probability that the deviation between our observed and expected is due to chance. So it's a very small probability that what we're seeing is due to chance alone, between 1 and 2.5%. So do we reject or do we fail to reject? Commonly we use a 0.05 as a common cutoff point for acceptance. If the p-value is greater or equal to 0.5, we accept that deviation we accept that the deviation is due to chance. So therefore we fail to reject the null hypothesis. If it is less than 0.05, we assume chance is not solely responsible and the difference is significant and caused by something else. And in this case, we reject the null hypothesis. So in our case, our chi-square was 6, and if we go back to our table here, here's our chi-square here, and we can see our cutoff here is anything below 0.05 we have to reject. And so we have to reject this. So we reject our null hypothesis. So again, we are saying that there has to be something else besides chance that's responsible for this. We don't know what that is. We might come up with some things. For instance, it's possible that maybe one type of the cats die before birth. Maybe there's a lethal combination of these alleles. Maybe a big B combination causes the cat to die before birth. In that case, our numbers are going to be thrown off. For a dihybrid cross, the difference could be that the genes are linked. So maybe the genes are on the same chromosome, and so therefore we're going to get different kinds of numbers because they don't independently assort if they're on the same chromosome. But this particular example is a monohybrid cross, so probably the best explanation is going to be this one right here. That one of the categories, big B, little b, or little b, big B, die before birth. All right, there are many other examples in your textbook as well that I would encourage you to look at. Okay, so in class today, I'm going to give this particular problem. And I'm going to ask you to work on it on your own, and then on Monday, or rather Wednesday, we'll go over it in class. All right, so let's summarize probability and statistics. Genetic ratios are expressed as probabilities, thus deriving outcomes of genetic crosses relies on an understanding of laws of probability. In particular, the sum law, product law, conditional probability, and the binomial theorem are going to be important for you to know. Statistical analyses are used to test the validity of our experimental outcomes. In genetics, some variation, as we've said before, is expected, and the question is this variation due to chance? The chi-square that we use will help us determine if this deviation is significant. Okay, that ends the probability and chi-square online PowerPoint slides. If you have any questions, please make sure you come see me.